Welcome students to MBBS Online Lecture 2021 YouTube channel. The topic for discussion for today's lecture is about immunity. We have completed our hemostasis in the previous lectures. I would give a link of those lectures in the iCard you can see above in this video. So let's discuss it. What are today's learning objectives? And the topic of our discussion is definition of immunity, what are types of immunity, a brief overview of acquired and innate immunity, and what is the mechanism of action of innate immunity. That includes the components of the innate immunity, the cell involved in the innate immunity, and what are the receptors involving in innate immunity. First of all, the definition of immunity, that is the body's ability to resist or eliminate potentially harmful foreign materials or abnormal cells. These foreign materials can be of any type, such as bacteria, virus, or any environmental antigen that is foreign to our body and is harmful, potentially harmful. So body's ability to resist or eliminate those harmful substances is termed as immunity. There are two types of immunity: innate immunity and acquired immunity. Innate immunity is a type of immunity that is present at a time of birth, while acquired immunity is develops during our lifetime. Innate immunity consists of organs, tissues, and cells. Such organ is skin. That is a major component of innate immunity. Innate immunity is also non-specific. What does that mean, non-specific? It means that it resists to all type of antigen, regardless of their specificity. Mean it cannot allow any substances to pass through it. While acquired immunity is a specific type of immunity, we would discuss the specificity of acquired immunity in coming lectures. So acquired immunity is also divided into two, that is active immunity and the passive immunity. Active immunity develops in response to an infection or vaccination. When a person is exposed to infection, such as any viral disease or bacterial disease, he or she may develop active immunity. When a person develops antibodies against such infection, we termed as natural immunity, natural active immunity. And when the vaccination is done, such as harmful foreign substance or any antigen is injected into person's body, and that may initiate an immune response and results in the development of antibodies against that specific antigen. While passive immunity is totally different, it develops after you receive antibodies from someone or somewhere else, such as in artificial, we are injecting artificially made antibodies such as immunoglobulins inside our body. So that may provide some defense against specific antigens. While natural passive immunity, the example is breastfeeding through mother. The antibodies are directly transferred from mother to baby. So this is type of natural passive immunity. What are the components of normal immune response? It consists of innate immunity and acquired immunity. What is innate immunity? I have discussed. It's a natural type of immunity that is mediated by cells and proteins that are always present and poised to fight against microbes being called into action immediately in response to infection. So they are the first one to respond to any infection that is non-specific and we termed as first line of defense. The major components of innate immunity are epithelial barriers such as skin, epithelial lining in GIT and in respiratory tract, which prevent microbe entry, while phagocytic leukocytes such as neutrophils and macrophages are the specialized cell type that that can phagocytose the accumulated antigen or foreign substance, while there are natural killer cells that directly kill those microbes and foreign substances. Also, there are several circulating plasma proteins, of the most important of which are the proteins of the complement system. We would discuss complement system, but I would give a brief overview of complement system. What is complement system? It is the complex of 20 proteins that are plasma proteins that work in a cascade manner and causes the production of membrane attack complex that, that directly attaches to the any any microbe, bacteria or virus surface and causes destruction. It is also mediated by antibodies. Here the components of innate immunity and adaptive immunity are shown, such as innate immunity. They are epithelial barriers. There are phagocytic cells. There are complement proteins and natural killer cells. While in adaptive immunity, the components are B lymphocytes and T lymphocytes. B lymphocytes result in the production of antibodies against a specific antigen, while T lymphocytes also causes the direct attack on the antigen by the cell. It is mediated by antibodies while it is mediated by cells. So we call it cell mediated immunity. We will discuss in the coming lecture. So adaptive immunity is normally silent and responds or adapts to the presence of infectious microbes by becoming active and expanding and generally generating potent mechanisms for neutralizing and eliminating the microbes. The components of the adaptive immunity are lymphocytes and their products. What are the components? These, these are two types of lymphocytes, that is B lymphocytes and T lymphocytes. B lymphocytes result in the production of antibodies that may directly kill the microbes or indirectly, such as activation of complement system. While T lymphocyte is a cell-mediated immunity, it causes the production of effector T cells that directly cytotoxic the microbes. There are two types of adaptive immunity. First one is humoral immunity, that is mediated by soluble proteins called antibodies that are produced by B lymphocytes, also called B cells. Humoral immunity is mediated by B cells that result in the production of soluble protein called antibodies, while cell-mediated immunity mediated by T lymphocytes, also called T cells. So there are two types of adaptive immunity. First one is the B cell or humoral immunity. Second is the cell-mediated immunity that is mediated by T lymphocytes. So innate immunity, what is the mechanism for action of the innate immunity? The components of innate immunity are phagocytic cells that include neutrophils, macrophages, and also dendritic cells. These all are phagocytic cells. So, and there are natural killer cells that, when stimulated or when active, can kill many of the microbes directly by releasing toxins and many proteolytic enzymes. Several plasma proteins such as activation of complement system. Innate immunity, phagocytes and dendritic cells and many other cells such as epithelial cell express receptors that sense the presence of infectious agents and substance released by the dead cells. One of the action or one of the function of our immune system or the innate immunity is the recognition of self and non-self antigens. So this recognition is made by expression of receptors that sense the presence of self or non-self antigens. So what are the microbial structure? What are the structure that are identified by these receptors are PAMPs, PAMPs, that are pathogen associated molecular patterns that may be bacterial products or viral products that are directly produced by those pathogens. Such includes peptidoglycan from the bacteria and toxins that are released by virus or bacteria. While DAMPs are damage associated molecular patterns, these are these are the molecules that are from the damaged cells. This include this include uric acid, the breakdown of DNA product, ATP that are released from damaged mitochondria, reduce intracellular potassium concentration because of loss of plasma membrane and DNA. So receptors of innate immunity are receptors that are present on plasma membrane detect extracellular pathogens. Such receptors are TLRs, that called toll-like receptors, while endosomal receptors detect ingested microbes that are bounded by vesicles. Example are NLRs, that are node-like receptors. Also, cytosolic receptor detects pathogen present in cytoplasm. So, the pathogen that are present in the cytoplasm of a host cell, the cytosolic receptors detect those pathogens. These all are these type of receptors. The examples are NLRs, node-like receptors. So, when node-like receptors are stimulated, they cause activation of inflammasome. What is inflammasome? That is a cytosolic multi-protein complex. That is a complex of protein present in the cytosol, which in turn activates enzyme caspase 1 that releases interleukin 1, that is the mediator of inflammation. As we have briefly discussed and in detail the inflammation process in previous lecture, we would know the importance of release of interleukin 1, that is the major mediator, chemical mediator for the inflammation. So these are the receptors shown, such as the toll-like receptors are like a hook present on the plasma membrane. These sense the bacterial products. What are the bacterial products? These are PAMPs, PAMPs and DAMPs. When pathogen is extracellular outside the cell, the toll-like receptor sense those pathogens. When the bacteria or any bacterial product is present in the cytoplasm, the node-like receptors or cytosolic receptors sense those antigens. When there is an endosomal formation of a vesicle around any bacteria or virus or ingested microbe, the toll-like receptor also present on that vesicle. We call it endosomal receptor. These are also toll-like receptors that sense the bacterial products present in the endosome. Reactions. What are the reactions of innate immunity? First one is the inflammation. Cytokines and products of complement activation as well as other mediators are produced during innate immune reactions and trigger the vascular and cellular components of inflammation. Mean when there is a inflammation at any site or the presence of the antigen or the foreign substance at any site where the inflammation occurs. So there is the entry of the neutrophil phagocytic cell macrophages that causes the vascular changes and cellular changes. We have briefly discussed or we have discussed inflammation in detail in previous lectures. So one of the reaction for innate immunity is inflammation. It causes induction of the inflammation, such as the activation of inflammasome that causes caspase 1 to release interleukin 1, that is also mediated for inflammation. Such receptors that sense the PAMs and DAMs, that are the microbial products that are sensed by these receptors, that can be peptidoglycan products of damaged cells or bacterial products. So that's the end of the lecture. We will discuss adaptive immunity in detail in coming lecture. So that's all for now. If you like this video, don't forget to subscribe and share it with your friends. We will see you in next video.